Today we will talk about infer function by motifs. Tatjana will not give her special lecture. I have to change that. Tobias will give that lecture, I believe. There will be a lecture from Andreas Schafferhans just before Christmas. And again, we talked about molecular biology. We talked about compute aspects of molecular life and the most accurate way to infer function. And we talked about how well we can infer by homology. Let's talk about motifs. Sequence motifs. So in fact, motifs, this is the protein sequence, so something long, right? And what we discussed so far is, can I look at two of these sequences and infer that two proteins have a similar structure or function? And Tobias showed you a machine learning device that in fact infers by machine learning through similarity. So it just takes a database of proteins for which you know the function. It sees is any of those similar to the sequence you start with. If yes, it uses that information in a machine learning device to infer a function. Okay? That's what we talked about so far. I also showed you how we can use that to infer particular aspects of functions, of cell localization or enzymatic activity. Now today I'm going to talk about short motifs. So motifs are defined as opposed to the entire sequence, something that's very local. And it could be this type of local, a stretch of residues in a protein. It could be the regular expression kind of local. Any idea how you would in biology possibly interpret? Why would it matter that a motif like that is matched? Why could that matter for the inference of function? So the protein sequence is this entire thing there. For the alcohol dehydrogenase in this particular case is 95 residues. And the whole thing has a function, and the whole thing has a similarity to something else. How could it be that a local, very short similarity could help me, or even a regular expression, which in many ways is even shorter? Yeah?
Because of evolution.
Yes?
Those sequences evolve, and during the evolution, maybe evolution decided to keep some specific part of the sequence because it has a function important.
So what could that be? What could that part be?
Like building blocks.
Mechanically. Yeah. Building block? So like a helix, you imagine? In structure, it would be, the building block could be a helix.
Maybe a binding site.
Exactly. It could be a binding site. So it could be, imagine that there is a particular pocket formed by a protein, and another protein binds into that pocket. And that particular pocket may sometimes, there's a so-called histidine triad. Those are 3 histidines that have to be at a certain spacing, and they recognize the substrate. So all you need is that there's not too much space between them. 3 histidines, not too much space between them. That's very much like a regular expression motif. And that would define that binding site. It will not define what else happens in that protein, but at least it will define some similarity. We could then say, okay, these two proteins most likely have a similar binding site. But now the question is, how could we find such motifs? Any idea? Any suggestion?
Not for similarity. Then we say we're looking inside the whole thing just for this small one.
But how would you find those? That's true, that's what we're looking for. Essentially what we're looking for is, for instance, a data set of these motifs, or just a bunch of those. So far I told you, no, it's trivial, right? I have a whole lecture today about motifs. So somehow this thing must work. But how can I establish? Let's find the first one, or the first 10, or whatever the number is. What is your idea how to find them?
We can do a multiple sequence alignment.
Yes.
Look for the small similar parts.
Yes.
And we can start with thinking about these as motifs and search for others.
Yes. So let's just assume. Let me repeat what David says. For those of you who are not that familiar with the biology in the background, so we begin with a protein. And in this particular case, I'm not writing the sequence, I just mean that is my protein sequence. I define another protein sequence that essentially aligns everywhere and looks very similar and has 99% sequence identity. There's just one thing here that is not right. So how do I find my motif?
You line another one?
Yeah, so everything is my motif? You said something. Let's find a local motif. The motif defined as where I have an overlap. And let's assume in this particular case, I knew even that the binding site would be in this region here. Let's assume it's a sequence consecutive region. And the binding site is in that region. How does that help me to find the motif? Somebody else. It's David's idea, and he's on the right track, but I'm exactly using the part of the idea that is wrong, just for the sake of being mean. So, how does it continue? Anybody else? Say the opposite. The opposite, sorry.
Can we make more comparisons with other proteins?
Yes, that we can. But let's just begin with the two. So what do you mean? Sorry. What did you mean by that? So you pull another third protein into this game?
Yeah.
What do you want the third? Yes. So we pull a third one in here. And I just don't have black anymore. Imagine this is black. That has 99%. So does that help me? And how does that help me in your idea?
What is the other one? Is it like full pattern?
Yeah, so essentially what I'm trying to say here is we have a protein, could be the ADH that I showed before, 90 residues or something like that, and I have an alignment of the entire 90-something residues, or the entire, say it's 100, and 99 of these 100 residues are identical, 99 are identical, that's the only part where I have a mismatch here.
So we match it with the 100 one?
Yes, we match. So all the residues except for this one here are identical. So we have now 3 sequences. They are absolutely identical in the binding site. I happen in this particular case to know the binding site, and they are absolutely identical. How does that help me?
I would say we take the whole thing first. So we have no
Okay, so now the obvious idea would be to in fact just take the whole region that is experimentally known to be the binding site and read off the letters that we have. What's the problem? Essentially I have identical proteins here. So I have no idea whether and I see that they are conserved simply because they are conserved everywhere, inside and outside the binding site. So if I just reproduce the same protein over and over and over again, I don't introduce new information. And it doesn't help me with my problem. What I need to bring in is some divergence. What I need to be able to say is, all right, we have a conservation here in the binding site, but not everywhere. If the protein is entirely conserved, there's no information content with respect to the binding site. Then I'm back to the old story of entire sequences, right? So, okay, let's assume that we started in the same way and now we have 66. This is 66%, and I read off the motif. Am I done? And say the motif would be, this is the motif that I'm going to write down as a real motif, just it's not really functional. Has to do something with function, is a GXX, GXXG. Now, the door is open. You can come in if you want to. So assume that is what I find. And again, now we have a situation where obviously I don't have full conservation anymore, where obviously I have X's in there, meaning I have regions that are not conserved, and in this particular case the meaning of the X simply is I have any of the 19 letters, any of the 20 letters there. But it must be 2, the glycines, the G's, must be spaced by exactly 2. That's the meaning of this motif, okay? Now, am I done? Again, we want to do that again and again, but let's work on one at a time. Let's begin to establish the first. I have this established. I know this is conserved throughout some set of proteins. I know it's a binding site, so I know it's somehow functional. And that is what I see in the alignment. What else do I have to do? Is there anything else I have to do? Or let me put it differently. Obviously, I do have to do something else because I'm fishing for some answer. Any idea what that could be?
I'm not sure if you can do that because when you have like 66% sequence alignment, but the protein has a different function, it might not have
that's a very important issue. So whatever you align, if you build your first motif, you better be sure that you can believe that in fact it has the same function. Let's assume that is the case. That's a very good point. That's a very, very good point. But let's assume that is the case. What else do I have to do? And it is aligned, so the answer has something to do with what you just said.
I think I'm lost a little bit.
Yeah.
What is the goal here?
So let's get back to what I said before. Initially, or the last weeks, we talked about finding similarities on entire protein sequences. I take the entire protein sequence and I find a similarity somewhere else in another entire protein sequence. Now I talk about motifs. Motifs are short regions. And those short regions make sense because they may reflect binding sites. They may reflect this protein here may have two different binding sites. But maybe this motif is related to one of them. And maybe what I find is not the same ADH1 with two different binding sites, but maybe I find one of the two binding sites. And that may be a way for me to learn something about the function by knowing the binding site. So what I'm after are short motifs. And the question really is, how can I find them? And the idea was, let's begin with alignments. Let's begin with a set of proteins here that diverge, so that do not all over have the same sequence, but that have some local similarity. And in this particular case, I experimentally know that is the motif. I postulate that is the motif, and I say the motif is this GXX, GXXG. And I've established that a couple of proteins that have a similar function have this motif. Now my question is, what else do I have to do in order to call this my first motif that works? Well, we have to establish that it does not hit in proteins that absolutely do not have that function. It's similar to what you said before. But now the idea is what I buy from this. So far, I built the multiple sequence alignment again using the entire sequence somehow. Maybe not the entire one, but a larger stretch. And I read of this larger alignment something very local. Now, the idea is that with this local thing, I can now go out into the world and find things I did not find with the entire sequence. So now let's go out there and see what I find with that motif and find a way to check that what I find makes sense. This happens to be a membrane motif. So if most of the proteins, or many proteins that I find, have nothing to do with the membrane, in this particular case, I immediately know the motif is wrong. If the motif had something to do with DNA binding, and I find a lot of proteins that are outside the cell that typically don't bind DNA, I know immediately that it is wrong. Okay? And in principle, this idea has been realized in 3 databases in slightly different ways. The first one is the oldest and also the mostly used one, PROSITE. Protein sites, and there you already have this word binding site in it, in the protein, right? It comes from Amos Bairoch, who in fact started a database, Swissprot, PROSITE, Exposé, Califo, at the Swiss Institute for Bioinformatics, and is one of the major people in the field, even so big that there is a special dole from him. Anyway, so again, the idea is exactly what we talked about before. We have the alignment, we see some local conservation. In this case here, we just have these 4 columns entirely conserved. I make sure that this is in fact a very specific motif. And I find this motif somewhere else, and I transfer the annotation. This is simply the idea that I just described. Now, the idea behind PROSITE was completeness first. So find as many of these motifs as possible. high specificity, so the motifs that you find should not be such that they cover things that are not membrane proteins, just what we described. And the word here is high because you cannot always make sure that nothing that you find is not right. Have a documentation and have a periodic reviewing. Now, PROSITE exists since over 2 decades. What do you believe is the most complicated one of these 4 goals?
Documentation.
Lazy hacker. You're not off, but I would not necessarily. So documentation. So you may be a lazy hacker. I'm a lazier hacker. I don't even take it seriously. Fair enough. Yeah.
I think the completeness, because of the model of the data.
We will get back to that. It's a very good point, and we will get back to that. But ultimately, for many tools that you develop, if they are supposed to do something real, we're talking here about grep. For instance, regular expression motifs, right? grep has been developed in the 80s, right? And it's a thing that you still use. It's incredible that these kind of simple algorithms stick. But it means that really you have, for grep it can work because it's very simple. For many tools it doesn't work and they have to be rewritten. But the periodic reviewing is in fact the problem. So the more the problem that you talk about, the completeness, is solved or approached, the more this becomes a problem. The larger the body of things that you have to go back to review. Discovering a new motif may be fun, but seeing whether it's still true is not fun. Because the only thing that can happen to you is that you find it was wrong. And that is not fun because you didn't remove one. And the database that you curate is getting smaller and smaller the more you do. And that is not fun to do. So this ultimately is one of the major issues. Again, that is the way it started here in 1991. And later then, however, in fact, completeness became a big problem. This issue of completeness became such a big problem that they tried to somehow automate that. What I showed you so far really was done manually. Motif by motif, yes?
Is even the periodic reviewing made by hand, like
yes,
automatically?
Exactly.
There's no way to make it automatic?
So yes, there are ways to make it automatic, and those ways were implemented. So I gave you an example on the previous one. There is this GXX-GX. In this particular case, all you need to do is you know exactly that it's not supposed to hit the membrane proteins. So you can predict membrane proteins, which we did in last semester. And then you can simply say, the moment I find membrane proteins out. That's easy to automate. The moment this is a motif that is more specific, histidine triad, you can find many histidines in proteins. So finding 3 histidines is meaningless. So is it, maybe at some point you say the spacer or the in-between, you can have only hydrophobic residues. And you find tomorrow a new histidine triad protein, is that then still true? And that you cannot automate. In fact, many you cannot automate. Some you can, and so they automated filter mechanisms that filter essentially, what is the simplest filter you could imagine? Automated periodic reviewing. When something goes, what is this? Imagine something goes extremely wrong. How can you detect that? It's very easy. And filters like that have been implemented. But think about can you imagine any filter that is simple? Again, remember, you have a motif. You want to make a motif that is very specific. What does it mean?
Has to be short.
Short, yes. No, you put the motif into the database, and periodic reviewing is about making sure that tomorrow this motif is still right. Give me an example how you can immediately show that something goes wrong tomorrow. Or imagine a case, or give me a case that you could check. That would immediately be the case of problem, red flag. A very simple red flag case. This motif sits there, the database grows. How can you immediately produce a red flag?
When this motif is not reproduced. So
when nobody finds it anymore.
Yeah.
But you still have the old sequences in there. That is certainly a red flag. But that red flag means you have a programming mistake somewhere in your regular expression, right? Because the same sequences with which you did the motif today are still in there. So if that happens, you're right. That actually is a red flag, but that is more like, let's call it an error in your program kind of red flag.
Okay.
And it's true. You need that. But I mean something that is not quite the error in your program, but comes from something that comes into the database.
Sequence is updated in the
Yes.
database?
Yes. So the database, currently the database of protein sequences doubles in less than a year. Doubles, it has currently 55.
Like, if you have old sequences, is it possible to
Yes.
update them and say, I need
Yes.
an error? Like,
Unfortunately that is the case, yes.
so that could be right.
Yes, David is right. So you got a good way to make David right. You're right. Good point, but that's not the one I'm fishing for. No, but look at the other extreme. So assume this week we have 10,000 edit, and suddenly they all
match.
Yeah, exactly. If you suddenly have a large set that comes overnight. Now this again, it could be two things: that suddenly somebody's sequencing all the DNA binding proteins, or most likely something went wrong with that motif, sour with that motif. So and you could essentially. You build up simple flags like that. That is the automated ones. And over the years, they found more and more. Philippe Burkhard from Lausanne, in fact, has automated the entire approach by using longer motifs. That is just a set of all the publications on PROSITE. The size of the list, the merge size of it, tells you that's very active research, and there's a lot in it, and PROSITE and PROSITE Scan is one of the major resources. Let's go to prints. Now, what else could you improve from what we have here? Again, we're trying to find binding sites. Any idea? We still stay in the same theme of we are going and build the alignments, we're looking at motifs. The idea of prints is, in fact, to connect motifs. To say, okay, this is a short motif, this is a short motif, they have to come together. I need both of them. And that is not really built into the PROSITE original idea. It could be in a regular expression, could capture that. But prints is really the idea to explicitly build that into the formalism of building the motif database. And there are, in fact, stretches of evolutionary conserved fingerprints. That's the idea there, and has grown, so the Homeo box would be several motifs come together. Here's an example for that. Blocks. Blocks now is another similar idea, but in some sense more extreme, and it comes from Stephen Henikoff, who produced the Blossom matrix. What's the background to this one here comes from the following observation. You see that this is a typical alignment that you see, where the green sequence is the one you start with, and these are all aligned sequences. The white places are regions where you cannot align the two. What jumps at you?
The sessions.
Yes, the white is correlated, right?
Yeah.
So that implies that we see regions where a lot is conserved, and we see regions where almost nothing is conserved. If that is the case, then that must tell us something about evolution, Henikoff argued. And that means let's just find blocks, regions that are entirely conserved. And these blocks, in fact, will carry some motif. That's the blocks database. Now, the beauty of that is very easy to do. You just align sequences, find conserved regions, and you're done with your database. Overall, this is the length in a family, that's the overall percentage. 100% of all the blocks are shorter than 40. And most of them, 40% here, are very, very, very short. That's the downside to it. The more divergence you add, the fewer blocks you will get. And that ultimately is also the reason why the blocks concept was stopped. But it was very, very important while it lasted, and it would still be good to make something like that work. But now here's another idea. PFAM, and the name stands for protein family. The goal is to build protein families. How would you do that? So we want essentially to build things where then once we have them, we know they are somehow related. And since we know that, we can now build a PROSITE in the future better. That's one rationale. But also another rationale is when I'm hunting for related proteins, I already have a database of these families and I can just go there, pick it up and see those are the related ones. But how can I build this database of families? And again, the idea is that I have proteins that are functionally related. So everything that sort of does the same thing is in one family.
Are the blocks in the same?
That could be the one. So you could just argue, you just build them by blocks. The problem with the blocks is they are so short that they are not really covering families. So that would have an atomized way of doing it. Yes, blocks was essentially the idea to do something such as this in an automated fashion for the entire universe. And it worked for some time, but then we got too much divergence. So how can we do it?
So we need to match more than two possibilities.
Mm-hmm. But my question, more than two or three or some, but how will we do that? Just imagine I charge you with writing the algorithm. What would the algorithm do? So you have a database. You have methods that align proteins. So you take a sequence and it finds everything that is similar to it. We discuss the issue with function in a moment. What can you do?
I mean, it depends how big you want to go. So
that's true. All proteins, or at least all proteins in us, or something.
but you need first of all some classifications, so, some family names which can either be solved manually by saying okay this and this and this.
Manually. Or visually, yes, one at a time. Okay, that's one solution. Any other solution? And since you guys are not really the experts in biology, you may want a more automated solution.
Just to find some patterns there?
That's what we did so far, right? But in some sense, so what I'm talking about now, you could use PROSITE, but now I'm talking, you want to, in fact, build a data set of all the protein families. PROSITE doesn't exist. Now PROSITE exists, and today you could use it. But actually there will be issues with PROSITE as well. But now assume that PROSITE didn't exist. What I showed you so far is PROSITE, but it didn't exist, and you wanted to build a family database. How would you do that? Manually, one at a time. But how could you automate it?
Again, we define a family on the basis of
it's a very,
we define a family.
It's a very, very, very good question. And I cannot answer that question before you suggest a method. Well, how could you? Let me give it back to you.
My idea was to use the sequence that had inside like the feature we are searching for. And
this is what I would call clustering. You two have the same idea.
Oh, okay.
There's a clustering of the idea in the room.
I want to teach you something.
Yes, but that I already answered. So assume there is no PROSITE. And PROSITE, remember the way I told you PROSITE evolved, is in fact one at a time. And we get back to that issue with one at a time in a moment. But that already, let's call it, was David's idea. So it's already taken. Come up with a new idea.
We want to match a bigger set, not one by one.
Mm-hmm. Again, we'll talk about it one by one.
If we don't
We
have
could use the regular expressions.
No, we cannot, because for that you need the PROSITE database, which you don't have. And in order to build the PROSITE regular expression, you have to do one at a time, and that idea is already taken. So, restart.
If we don't have any base class,
Exactly.
then the only thing is like just to visualize and see.
But have you taken last year's lecture?
No.
Yeah. Of you, anybody has taken last year's lecture?
Yeah.
No, of this side here. No, nobody? You have. So the only victim who's eligible. So essentially, the idea is you do an all against all comparison. You compare families against families. But, okay, let's get back to David's idea. So manual. Okay, we have 20,000 proteins in human, okay? How many families do we typically expect there? Maybe 10,000, maybe 5,000, and you want to do that manually? How do you imagine this? How do you imagine this? You come to work in the morning, you do an alignment, and in the evening, maybe you have one new entry, and this way you do the whole world of proteins?
I'm pretty sure that's how it started.
Yes, that's exactly how it started. That's the person who did it. 10,000 over the last 20 years. He has done PFAM, now he has added RFAM, that's a little bit more automated, and actually is partially cloud done, or contributed a lot by people in Wikipedia. And Alex just stands up and says, I'm a stamp collector, that's what I do. My goal is to annotate all protein families in the world. And he has pushed over the last 20 years essentially 10,000 or something in that ballpark. And that is precisely what he did, with the help of tools. And in fact, once you have this family here, here's an example. It's not an example for a family, I'm sorry, I should have put the example for a family in. Once you have an alignment, a family, then you can just construct a motif, a hidden Markov model for the family, and take that hidden Markov model, and with PFAM, in fact, every record in PFAM has a family and a hidden Markov motif, which you can use to run to find new sequences.
I'm sorry, but how long did it take him?
20 years.
20 years to manually do this.
Yeah. And they are not done.
And how can you be sure that it's right what he's doing? I mean, it's like you're spending 20 years of your life annotating programs, so you're not sure what you're doing is correct.
There's a very, very simple answer for that. Oops. Everybody loves it. And the people who quote the work are the people who are experimentalists who verify this. That they find the families, they find that this... But here is one problem that I omitted, and that's a very important one. So, say this is one protein, and it has the red and the green, let's call it feature. Here's another family, and here's another family. How can I make sure that what I annotate has the same feature in this example?
So what you're saying is the families are overlapping? Or?
Mm-hmm. The families are overlapping. I believe it would be better if I had a little bit of green here or something like that. So the answer that I'm trying to find here, you have to cut. And in fact, that is the major problem here. So that we in fact have regions that are domain-like, or shorter than domains, and that are not really motifs. So PFAM finds a way between taking everything, taking the PROSITE motif, is somewhere in between these two extremes. Yeah?
Could you repeat please?
So the idea of domains is simply proteins consists of unit. And you can in some sense imagine that each unit has to do something with function and structure. And you can imagine that in fact these units, since they have their own function, the red unit here, they are duplicated in evolution. So you will find possibly another protein with only the red unit, or with a red and an orange, or with a red and a blue, or also with a red and a gray.
Because in the end the red is a function. So
the red is the function in some sense. And then the point here is, if you knew this function clearly, in the functional region, then you would know exactly where to cut and which region to yoke. But typically you don't. Staring at these alignments and becoming an expert at that, you sometimes can see where you cut. And there are algorithms for that I went into last semester. I will not get back into that. But there are some tools that can help you to delineate these regions. Ultimately, you're looking for regions that are such that inside of this PFAM domain, everything is sort of conserved. And still don't go to blocks, still don't go to the shortness of blocks, and don't go to the shortness of a regular expression, but something that is sort of 50, 60 residues long. And that most likely is the kind of thing that we assume reproduces in evolution. That's the goal. And that, in fact, makes it even more challenging to get these regions. There are several competitors, and typically these competitors have a high overlap, as is shown here, TigerFam and SMART. This is an old version here of 2003, so it's over 12 years old. But you see sort of the relative sizes and the overlap. The original content really is mostly in PFAM. This one shows you the length of the domain, and essentially it shows you, if I gave you enough time, I don't want to give you enough time because I don't want you to learn it and to be worried about the detail. Essentially, PFAM is a little bit shorter than what we would call a structural domain. Now, what else could we do to discover motifs? By the way, the all against all issue, and I again forgot to put the slide in, there is a tool from Hebrew, Protonet. And Protonet, in fact, finds an interesting way of clustering using the entire sequence. And the way they do it, from clustering the entire sequence, is they still see relations by users going through the network of connections. That's an absolutely cool, unique tool that I should have spent more time on. I don't. Let's get back to the issue of motifs. So we're back to, rather than sort of domain length, something that's more like a regular expression, something that is shorter. How could you find them? We talked about the manual way of doing it. We talked about all the ways that if you would have studied more biology, you could do. But you're computer scientists. How could you do it without knowing too much about biology?
So what's the problem with this finding all?
Takes a long time.
Huh?
Takes a lot of expertise and a lot of time.
For the course is hard. Okay.
That's one problem. I mean, again, so it's the version that evolved historically. And it evolved historically because 20 years ago, today we know over 60 million sequences. When these guys started, we had so few that they could still hope they would cover the universe. If Alex Bateman started today with PFAM, I believe he would be so scared that he wouldn't start. But then the database was so small, and we assumed that the universe is much more limited than it turned out to be. So I don't believe today even computational biologists would start with this again. It grew, it's an extremely valuable resource, and it was the right way of doing it at the time. But if we had to restart today, and actually other people, even at the time, did it differently, partially because they had a different perspective that is closer to yours. So I'm going to show you 3 methods that are more math, computer science based. If I gave you these words, does that give you any idea? The middle one, absolutely not.
Maybe I have heard the word, but I don't know.
Oh. There are many languages that you use this. In French it means the same. But that has nothing to do with nothing. But this could give you an idea, and that could give you an idea.
It's more frequency-based approach.
Exactly. So could you imagine that?
So we take the data we have,
Yeah.
we analyze for frequencies in specific regions, and then we filter.
Yes, in principle that's exactly right. So Chip Lawrence developed this, the expectation maximization Gibbs sampler for motif discovery, and that was a science paper in already 14 years ago. 21 years ago. And essentially you optimize, you maximize the signal that you have in a bunch. You get a bunch of sequences that somebody tells you have to do something with each other. And you simply align them such that you find the most important motif in that set. And most important always means you have a region that doesn't have the motif and a region that does have the motif. And you just superpose them. Okay? Taking the alignment. Reading off the alignment, you look at the most significant region. You could imagine it as a K-mirror. The problem with the K-mirror, however, is that the K-mirror is consecutive, and regular expression motifs are not. So you need to move away a little bit from the K-mirror. So the algorithm is a little bit more complicated. E-motif, and again, comes from Douglas Brotluck and Neville Manning, who, by the way, is now at Google, and did all kinds of other amazing things at Google. Now that idea really is to use a tree, a graph structure. So again, you have an aligned tree. In this tree you find regions that are similar, and those regions tell you something about where the local similarity is. When you look at the alignment, you will sometimes see, okay, this residue is conserved everywhere. And this residue change here is the reason why that is clustered somewhere else. If that is clustered somewhere else in terms of a tree, then that is an important residue. And that's the idea here. Use the tree structure to identify motifs. The third idea really is meme, and that comes from Timothy Bailey, and that in fact advances the Gibbs sampler in two ways. On one, it just takes unaligned sequences, so it does not insist on the motif being there in the alignment, and it allows you to throw things out, meaning that you are given a set of 100 sequences, and meme can say, well, the motif is only 97 of those. You were wrong in giving me the 100, but those three, really, the best motif I find is such that 3 of those 100 don't have that motif. And that simply is done by a machine learning type of way. The moment you have this motif, that's the meme idea here, then you build a motif database, and then you get to the next point that Prince, in some sense, addressed, but now you do it more electronically, where you have to connect motifs. Where, in fact, an isolated motif is not helpful enough. So now, the complication, can you see what the complication of motif combination is?
It could be a lot of combinations.
Exactly. The problem is always, you have to really, really carefully get the statistics right. The more possible combinations you have, the more you will hit something at random anyway, and then again, the question is whether that is meaningful or not. So getting the math in this right is extremely tedious. And that is in fact why this has also, again, taken over almost 20 years. Let's talk about a different type of motif. Localization motifs. And the motif-based inference of subset localization. The work here comes from Rajesh Nair. Remember, I told you number of residues aligned, percentage sequence identity. Blue are proteins that have a similar subset localization. Red are proteins that don't. And there are these extreme cases here where you have over many, many, many residues, the entire protein, they're very similar, yet one has a different subset localization than the other. How can we picture that? One way to picture that is the following idea. That's the nucleus, that's the cytoplasm, that's the two-folded membrane. There are proteins here in red. One is called importin and transportin, names are not important, that recognize an NLS, a nuclear localization signal, which is a very local thing. So that blue thing here sits on a protein. The importin binds to that signal, shuttles the protein through the nuclear pore into the nucleus. So now you can immediately see, even if you're 99% identical, if you have a short sequence motif, one has the motif and is imported into the nucleus, the other one doesn't, it's not imported into the nucleus. In fact, experimentally, you can just put 5 residues on a piece of gold and it will be imported. Those 5 residues are recognized by importin. You kill the cell with that, but this is a different story. You have a very golden cell, it's great. Anyway, so these motifs are really local. Now, there are 2 types of motifs. There are sequential motifs and motifs that you see only on a conformational, that you see only on a structure. What I mean is here, we'll get back to that, is a signal peptide, is the motif is in the red, so the thing is folded, the motif is there, the thing is unfolded, the motif is there. Sequential. The conformational motif is such that you see these 2 binding sites here, and they in fact make sense only the moment they are folded in 3D structure, and you see that they come together. And suddenly the motif hits at you. This is the kind of thing that I talked about before, the histidine triad, when you have a binding site. So the spacing actually is important in 3D. And you may not, when it's unfolded, unwound, you may not see it. Now when we started, the only motif in PROSITE, there was one motif in PROSITE. that had 90% accuracy, so 90% of the proteins that mapped that motif were known to be nuclear. And of all the nuclear proteins that were known at the time, 3% had that motif. And we wanted to add motifs to PROSITE. That essentially was our idea here. Following up on, in some sense, your idea, PROSITE had worked on all kinds of motifs. In fact, the way PROSITE grew is simply by publication of experimental information one after the other, and they did not look in a certain direction. Here we looked in a particular direction, nuclear localization signal, because we knew that this is very local. So the first thing you do, if you want to find new motifs, the database doesn't have it, what do you do? Google, right? You find literature. In the literature you find that. Red means positive charge. Does anybody in the room have an idea whether positively charged residues, so looking at this, you see a lot of red, right? Do you believe this is typical for proteins? Are they that charged? No, absolutely not. In fact, charged residues, there are only two really completely charged residues, arginine and lysine, and 2 of 20, that already gives you an idea, and they occur typically roughly at that frequency. So it's 1 in 10, so less than 10% of the residues are charged. And some of them are negatively charged, but this is the situation here. Now, how could you now turn this into motifs? One simple way is you simply take each one of those, build your data set, and are done with. There's a problem with that, and it's the same problem that we encountered before. What is the problem with that?
It would match the main time.
Yeah, exactly. So they are published by experimentalists, but they don't know the database. What they try is really they mutate residues and they see that this protein is still there. They don't try, is that the same motif that you find in the database somewhere else? This is why you're a computational biologist. Or you are not, but this is why you use computers and you bring your expertise in the field. Here are, by the way, some other motifs. So what you have to do is you have to build 2 data sets. One data set of proteins that you know experimentally are nuclear. One data set of proteins that you know are not nuclear. And you simply have to see that the motif, a good motif is such that it matches 2 things here that are not similar to each other. Back to the 99% story that we had at the beginning of today's lecture. So you should have things here that differ from each other, here as well. So you want the motif to match in 2 proteins. And you don't want it to match in proteins that are not nuclear. Okay, let's assume that you have a motif. One, you pick the first, you go one by one, you pick the first motif, and let's use this motif here, DNA helicase motif. Let's assume you match it only to that one protein. What can you do? If you want to capture more than one protein, how can you make it less specific? Exactly. Now, how would you make it shorter? How would you decide? So if that is the experimental motif, how would you make it shorter? Remove this, the first one or the last one? How would you decide that?
We cut the part that doesn't match.
Exactly. You cut the part that, well, actually, sorry. That's a great idea, but what I would do, in fact, is build an alignment of things where we know that they are similar. And then first be guided by that. And then what you say, because what you say is an extreme way. But anyway, either way you do it, your way isn't maybe a faster way. Maybe you should have done it that way. This is certainly hitting it harder. And then you always have your control mechanism by seeing if other things that are not nuclear match it, you make it longer again, or you change it in the wrong direction. And this way you iterate through that and you build up a set of motifs that in fact have a much higher coverage. in fact, then you use that to build a larger set, and the larger set is chosen that it doesn't make a mistake, and it has a fairly high coverage, and it worked very well. All of these motifs, however, were again sequential. Now we try to do the same thing for something where we know sometimes target motifs are there, and that is what Kraszinski did for the retention in the endoplasmic reticulum Golgi apparatus. Let's not go into the detail. The message is here, he worked on it for over a year and did not find a single motif. So in that case before, I quickly showed you the number of over 200 motifs that worked in this way. He didn't find any. So everything that had been described in the literature had the issue that it was not specific enough. And we could not make it more specific, or we could not make it more specific without losing the fact that it will match only one protein. So all we ended up with are motifs that had a certain probability. None of them was telling. Together, in some cases, they worked, but that, in some sense, is the example of a dead-end project. That was not predictable before we started that, because in fact that was sort of nuclear localization signal everybody knew is one of the important motifs, and I will get back to that story. And the other one were these retention motifs, and they just didn't work. And ultimately the reason why they didn't work is because experimentalists did not make this test of trying the database to see whether they hit to many other proteins as well. Now, it is a challenge to automate this sort of approach and to unify and to find very distant related things. We should be able to find structural motifs, those conformational motifs. So far, very few of those have been found. But there are many open challenges. One is because we don't find them. One is because we don't know structures for everything. We find binding motifs. That problem is solved. But the motif in this particular case that tells you about subcellular localization, we're still not far enough. We have, the idea really of motifs is ultimately the mechanism is a zip code kind of mechanism. So there's a protein that recognizes a zip code, and this way you get you transport it throughout the cell. Today, of all the motifs, if that really were the predominant process, for most of the motifs, we absolutely don't know what they are. So maybe they remain to be discovered. Or maybe there's an additional complication in their story. So I told you that importin is recognizing this motif here. Now, how could you imagine that another protein, this head protein here, is imported to the nucleus without having a nuclear localization signal? And still somehow adhering to the idea that everybody is regulated by zip codes. Can you imagine?
It has the zip code already inside.
No. That's a great idea. No, my head doesn't have anything inside. My head is absolutely empty. So that part doesn't work. No, what I meant is, imagine this one binds here. That has the zip code. That is recognized. And if that is bound to my head, and this is bound to the importin, then the whole thing is carried through the nucleus. So essentially by binding to another protein that binds to, how could we estimate whether that in fact could be the answer to the quest? So we look at all the nuclear proteins. I showed you, unfortunately the answer that I gave you is no longer valid, but when we did this, we believed that we could cover 43% of all the nuclear proteins. How could we see whether it could, in principle, this explanation could cover everything? What I need to find is for all those that have one of these motifs, what they bind to, and whether that sort of explains what we know. Or at least as long as that gives a significant jump from where I am, then I have a hope to be in the right tendency. Unfortunately, we couldn't do that at the time because we didn't have enough interactions known. This is something that we could possibly redo again, and that is in fact bringing us to another theme. The annotation of function by association, which is essentially exactly this sort of idea. You're in a group, you are seen in a group experimentally, then you are supposed, we expect that you do something similar. Yeah, let me give you an example. This is an expression data array. So typically, in the simplest way here, red means it is not expressed, green means it's expressed. So typically down here you have conditions, so you do different experiments, you deprive the cell of sugar, you put it under pressure, you put it under heat, blah, 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 blah. And each row is one particular gene or protein. And you see some patterns showing up. And from correlated patterns, so everything that in heat is always green, you suspect, there's a condition under which all of these genes are switched on. They are expressed, they do something, they're suddenly needed. Why are they suddenly needed under one particular condition? Well, maybe that is part of what you would call a pathway, a whole process of activity toward helping the cell to survive heat. So then the idea simply is, find all of these correlations, and from these correlations, look at things that are related in some functional sense. This way, you cannot say that these two proteins, here that at least over some time have the same behavior in terms of red, green, of a bunch of conditions. You cannot say that they are two of the same enzymes, not at all. But you can say that, or you can suspect that they are part of the same process. One is the enzyme, the next one is the next one in the line of things. This is one way of doing it. Here's another way of doing it. And that is similar to what I just said before. So this is a view of protein interaction networks. And in these protein interaction networks, again, so if you now followed this and pulled out all the things that sort of interact toward one network, it's the same idea. You will get things that are related in a pathway, not things that have the same Go number. Sometimes, however, it could be that alternative binders, other proteins that bind to this, in fact have the same function. It could be that this protein here is cleaved by two different proteins. So two different proteins take the ATP away or do something else for this protein, right? Or it could be the other way around. It could be that this here is the cleaver, is the kinase, as these cleavers are called, that sends a signal. Typically, kinases send you put a protein onto a surface, you cleave something away, and then the cell recognizes this open point and does, for instance, import that protein. That's why kinase activity or signaling are so important. So you could imagine that this is the kinase, and it has different substrates, different targets, different proteins that it cuts. In fact, this is what kinases do. But you immediately see that from the network structure, you cannot quite read off this is that Go number. But you can read off associations, right? And in this way, again, you can somehow see similarity in function. The problem with that is you have to find a way to differentiate between functional and physical interaction. So the story, and I will get back to that later in the lecture, so A activates B, activates C, activates D. In a circle like that, in a cycle like that, on a path like that, A and D would be co-expressed, simply because they are all part of the same reaction, but they would not interact with each other, right? So depending on what you show in this network here, whether in this network in fact the connection means that there is a physical interaction or just an association, you get a very different interpretation with respect to the function. That's one issue. The other issue is, in fact, we need a larger coverage, and we need to be able to predict aspects of function. We can, in fact, predict subcellular localization, where the protein is. We can predict cross-translation and modifications. We can predict functionally important residues, and we can predict interaction sites, and we can predict to some extent protein interactions. Those are the things about function that we can predict without knowing anything else. Tobias gave you the meta student that predicted functional numbers, Go numbers. But this prediction was possible only because there's something that you looked up in the database. But somebody had done some experiment, and you could relate that experiment to that particular sequence simply through a motif, through an overall similarity in the sequence, and themes like that. But you could not predict it without that lookup. Those are all methods that gain information from a database, but for which it's not one lookup. It's machine learning. There's a sort of more generic motif underneath. And that's what the remainder of the lecture will be about. I will have one more detour. This one says the next is prediction of subcellular localization. Yes, so then that will be the next. So I will skip the other one. So the next stop here really is the prediction of subcellular localization as the first example for a prediction of something that really, where you don't need any other information. And that will be on Thursday, the lecture. Next Tuesday, there will be, on Thursday I will give a whiteboard lecture. And on next Tuesday I will simply repeat the same story with slides. On Thursday, Tobias Hump will talk about another prediction method. Yep. That is it for today. Thank you.